Today, we're presenting how NU GoKidney's Therapeutics Design and Development Core can help your research to help you get to know our core resources and experienced experts in a variety of areas such as novel cellular therapeutics development, virtual drug and biologics design, high-throughput screening platforms, and nanotechnology-based precision targeting. So, after today's webinar, you'll have learned about NU GoKidney and its mission, be able to describe the Therapeutics Design and Development Core and its services, identify ways Core B can help your research, and also learn how to work with Core B if you have a project that's a good fit. Just a little housekeeping before we get started. Today's session will be recorded, and we'll send you a copy of that recording. There will also be a short 3-minute survey after the webinar, so please take a moment to share your thoughts and help us improve and select topics for future webinars. We'll also have a few minutes for questions after each presenter and also at the end. So if you have questions during the presentation, please type them into your Q&A box within your WebEx control panel. So now, without much further ado, I'd like to introduce today's presenters. We have with us Core B's director, Dr. Jason Wertheim, and 3 of our core experts, Drs. Irawati Candela, James Matthew, and Carlos Vanoy. So now I'll turn it over to Dr. Wertheim to tell you a little bit more about NU GoKidney and its Therapeutics Design and Development Core.
Well, thank you very much, Ashley. It's great speaking with you all today. We're excited to tell you a little about the work that we're doing at NU GoKidney, and specifically within Core B, which is the Therapeutics Design and Development Core. I'm going to go over a couple of slides with you all just to give you a high-level view of the core and how to get engaged with some of the technologies that you'll hear about in today's webinar. The mission is to promote and support innovative research leading to the discovery of novel nephrotherapeutics that will transform and prevent kidney disease and eventually lead to cures. And so you see in the graphic here, there's a Core A that focuses on preclinical models, Core C on clinical and translational study support, and then where we fit in is Core B, which is kind of the bridge between the two in designing and developing therapeutics, sometimes based upon targets that are defined in preclinical studies that eventually will translate into Core C. What do we offer? We offer 3, the NU GoKidney, rather, has these 3 biomedical research cores I just described. Some of you know that there's pilot and feasibility funding. There's an open RFA call due in about a week to implement pilot and feasibility studies. It really utilizes some of the resources within these 3 cores. And we're happy to be a part of that in Core B. So if there's any technologies that we talk about today that's fitting in with some of the potential pilot project that you're interested in, please reach out to myself or the participants in today's webinar. We also provide some enrichment and training opportunities and also community programs within NU GoKidney. So Core B specifically is to establish efficient platforms to support and develop and validate a pipeline of chemical and biological cellular therapeutics to treat a wide spectrum of kidney disease. And so what you're seeing today is really just a sliver of the different cores and resources that we have in Core B of NU GoKidney. We're planning to have a series of these webinars that will progress throughout the summer and fall and highlight some of the different cores that are under the Core B umbrella as an example. So what we're showing today is not the limits, but just a small taste, if you will, of the different technologies. And they can focus on small biological therapeutics or cell-based methods, and also nanotechnology, which we'll touch upon a little bit today and probably expand upon on a future webinar. So how to get started with our core. We have a series of, we have core directors. So I'm the director of Core B. Evan Scott in biomedical engineering is the co-director of Core B, and we have a series of Core B navigators that you can find on the website for NU GoKidney. And so up here listed on number 1 is the website, www.nephrohub.org. And that outlays a lot of different resources within NU GoKidney, and you can specifically go to Core B and find our navigators that can help you through the process of developing small molecules. To get engaged, we would like you to fill out a REDCap form and request specific Core B services. That way we can track requests, and you can schedule a consultation with the appropriate navigators that I've mentioned earlier. And we can help you develop a plan and guide you through the next steps of getting your research and technology development underway. So with that, we'd like to kind of segue to some of the individual resources under the Core B umbrella. And so we have Dr. Anki Candela. She is the Assistant Director of the Developmental Therapeutics Core of the Chemistry of Life Processes Institute, who will tell you about how she shares her expertise in screening and developing targeted drug delivery for novel therapeutics for kidney disease with specific Core B users. So with that, I'll transfer over to Anki, who will talk about the Developmental Therapeutics Core within Core B.
Thank you, Jason, for a very kind introduction. So let me go through here. It is my great pleasure to actually be giving a web seminar today as a part of Core B of a kidney center. And let's go start with what is DTC and what is this core doing? So we are a core that providing a preclinical support to principal investigators. Mostly we are focusing on in drug development, usually just one stage before clinical trial. And if you look at in the personnel, it's me and Dr. Fraser Aird, and as well as Ivana Stapien that's assisting the PI. I directly report to Dr. David James, and like Jason mentioned, that we are under umbrella of direction of Tom O'Halloran. And what are we doing? What kind of services that we're providing? So this is a bit busy slides, but let me go through one by one. So we're providing efficacy studies in different models. So that's why I'm so excited that we are a part of Core B of a kidney center, because we would love to actually test a lot of probably kidney models. In the past, we usually work with an oncology model where we work with xenograft model, patient-derived xenograft, syngeneic, and the implantation can be subcutaneously implanted or orthotopically. We also growing PDX so that we can characterize the models so that the principal investigator can use it for screening their novel drugs. We also help PI to test in vitro experiment like viability test, proliferation assay, migration, as well as a combination index from 2 or 3 different drugs. The one that is very unique for us, we also is capable of performing toxicology studies, including maximum tolerated dose, where we can actually investigate what's the dose range of a certain or a new drug. We complete repeating dosing for 7, 14, and 28 days. We do exploratory PK, where we can understand characteristic of the drug, especially the clearance in the mice as well as in the rats. Recently, we were being requested for running a rabbit study for these 2 different models, which is macular degeneration and angiogenesis. It's very, very unique because we have to tailor a new iCook protocol for them, and we have to consult with FAT to make sure that the study is being completed in a smooth way. We also do a la carte treatment like IV injection, IP injection, sub-Q, retroorbital, or placing OZ pump inside the animal cavity, like peritoneal cavity. We also just recently being requested to run a breeding for example, reproductive toxicity study. So after we run the toxicology study for example, repeating dosing, we actually breed the animal so that we understand like whether there is a pup that being produced after receiving a certain drug. We also just run a cachexia model for Dr. Teresa Woodruff in collaboration with a company in California. We also do some implantation of devices, and I will show you an example of that. It's very unique, and we can actually measure kind of oxygen, a volume, a temperature in the non-invasive way. And then the last one, we also coordinate with other cores to achieve a final goal of the studies. Last but not least, we also work in collaboration with other cores to actually run immunization of the animals to get peptide or antibody with RPPC and peptide core. Next slide. So how we, if a PI is interested in working with us, usually they can contact us via email or give us a call. And usually we discuss in details what's the goal of the project. It can be for just a simply grant application. We need preliminary data, or it can be for publication, or it can be for IND application to submit to FDA. And we usually come up with a cost estimate and draft protocol. And we make sure that the PI approve that. And usually it's back and forth just to make sure that we agreeing to both of the parameters, meaning cost estimate and draft protocol. We usually adjust if the PI doesn't like the cost estimate, we adjusting it appropriately to fulfill the budget. Once we finalize that, we have a lot of iCook blanket protocol that we could add addendum to, so the PI doesn't require to write their own iCook protocol. And we submit the addendum, and then the iCook reviewers will review them and coming back usually with the question. After 2-3 weeks later, we got an approval. And once the approval is received, we can start an in-life study. Of course, we need to purchase the animals, purchase the consumable. For example, if the study requires 4 to 6 weeks, then we run to the treatment until the end of the study, and then we usually send a report and then statistical analysis and the raw data to the PI. At the end, usually there are some analysis ongoing for the study, and so sometimes we work with mouse pathology core, for example, to give histology data and processing the tissue. We also work, for example, with high-throughput core just to get the molecular analysis ongoing. So we work quite with a lot of cores to coordinate to achieve a goal of the study. The next slide is an example of a pharmacokinetics study in collaboration with Dr. Stoddart. Here in the study, we actually try to prolong the ibuprofen effect in the formulation. And in this case, we're using a cyclodextrin metal organic framework, or CD-MOF. Before we actually did pharmacokinetic, we tested the formulation first in vitro, and it seems like from in vitro studies showing that this formulation did not provide cytotoxicity to the normal cells, so that we know that at least we're not going to kill any cells in our tissues. So if you look at the graph here, the first sample that we tried is ibuprofen as a salt form. The second one is a physical mix between the CD-MOF and ibuprofen, and the last one is the conjugation between the two. And if you look at the Cmax here, you see that surprisingly, the physical mixture between the two compounds is actually providing the highest concentration. Although if you see overall of the T maximum, all the formulation providing the very fast onset of a drug, meaning that it's available for the body to be absorbed or to be available for the drug within 10 to 20 minutes. And if you see it under the curve within 0 to 4 hours, the physical mixture is also providing the highest bioavailability of the drug. For prolonged AUC, you also see that physical mixture is providing the highest concentration. But if you see the half-life, the last formulation, which is the conjugation version, is actually providing the longest half-life, meaning it dropped longer compared to the other formulation. The second slide is providing an example of toxicology study in collaboration with Dr. Sui Huang. So in this case, we work with a small compound named MIN, and this is a derivative of aminomulfide that is the drug that is very toxic but is very good for treatment of hepatocellular carcinoma. And if you see here in this figure, we're actually testing this drug at 75 mg per kg via IP, and we evaluate the mice within 48 hours. I mean, basically, we euthanize them. And if you see the body weight, there is no difference between the vehicle and the mean. And then with the hematology panel, we see that there is also no difference between white blood cell, lymphocyte, and red blood cell. We also usually weighing the organs, especially the major organs. And down here, we don't see any difference between the vehicle and also the mean compound. And the most important thing is actually enzyme that coming out from a liver. And we don't see any difference between the two treatment as well, meaning the control and the compound. The next one, we also work with a nanoparticles. In this case, it's gold nanoparticles that conjugated with aptamer. And we would love to understand a biodistribution of these nanoparticles. We have 4 different concentrations that we tested. And from this, we learned that within 2 days, that the accumulation of female rats is higher than a male rat. And the accumulation is higher in 14 days compared to 2 days. So we can also learn about not only the toxicity of the nanoparticles or small drug, we can also understand the biodistribution or where those small compound or nanoparticles accumulated in organs. The last but not least is, like I mentioned, we work also with the device. In this case, we work in collaboration with Dr. John Rogers. And this device is actually measuring the oxygen. And for example, in this case, is measuring the oxygen in deep penetration of a mouse. Don't ask me about the device because I'm not a device expert, but their team is very capable and very expert in this field. So we work together, come up with the right model and the right condition for testing the device, and we usually collaborating with the imaging facility to understand the location of the device and as well as the function of device after we implanted it to the animals. So I think that's from me. Thank you for listening. And if there are questions, feel free to ask, and hopefully I can answer the question. Thank you.
Well, it looks like we don't have any questions as of yet. Okay. We'll let it marinate a little bit, and maybe at the ending Q&A we'll have some.
Sure.
So now we'll turn it over to Dr. James Matthew, who is the Director of the Immune Monitoring Core of the Comprehensive Transplant Center, to tell you about his work with NU GoKidney investigators on cell-based therapeutics.
Thank you, Ashley. Is it moving?
Yes, it is.
Okay. The mission of the Immune Monitoring Core within the Core B of the NU GoKidney is to provide consultation and support services for developing cellular therapeutics targeting kidney disease-specific pathogenesis. This involves protocol development before GMP production, transfer of the technology to the GMP, product authentication, and product quality assurance and quality control during the production and during the clinical trial. Then, during the clinical trial, serial immune monitoring of the subjects before and after product usage. Coming to the logistics, these are the how can we go about doing this. Initially, there will be a consultation with the Dr. Spertheim and the director of the Matthews Center for Cellular Therapy, that is MCCT, the Northwestern's GMP facility, and as well as with myself. Then we can design a comprehensive protocol for the product manufacture, followed by a small-scale manufacture either in your own lab or in the immune monitoring core. Simultaneously, we can design and develop assays for the phenotypic and functional testing of the manufactured product. Come on. It's not moving. Sorry, it's not moving. Oops. I'm sorry, having difficulty in advancing the right slides. Okay. Coming to the product authentication. Within these main functions of the immune monitoring core or the Core B, one of the functions is product authentication. This consists of a set of in vitro and in vivo assays applied to the dry run product. You will see about that in the next slide. And these assays may include, as shown in this slide, the turnaround time for getting the assay results will be minimized, and acceptable cutoff for quality assurance will be determined. Then also we can provide immune monitoring core, immune monitoring assays of the patients or the subjects after the treatment or serially during the treatment. And this slide shows some of the cellular assays that are currently available in the immune monitoring core. Also, we can develop additional assays if need be, as well as we can provide molecular, proteomic, and genomic, as well as animal study models in collaboration with other Northwestern cores. Let me give you an example of the couple of examples where the immune monitoring has provided services for cellular therapeutic trials. This slide shows a phase 1 safety trial of Treg adoptive cell transfer therapy in living donor kidney transplant recipients. The design was a single-center, open-label, non-randomized trial with the objectives to determine the safety of using polyclonally expanded Treg therapy, investigating whether TRACT leads to transplant rejection, allosensitization, or non-specific immunosuppression. We also performed limited immune monitoring. The methodology of the phase 1 trial consisted of dose escalation of expanded autologous Tregs in a single experimental arm, giving the recipients 0.5, 1, and 5 billion cells per subject, and we had 3 subjects in each tier. We published these results last year, and the primary outcome measurement was patient and graft survival at 1 year after transplantation. Thank you. Next one, please. This slide shows some of the product authentications and quality assurance, quality controls steps we have taken. Section A shows the GMP expansion protocol. The section B shows we have obtained the cell numbers obtained in the 9 Treg products that we have expanded. Section C shows the phenotypic scheme we used for analyzing the CD4 positive, CD25 positive, FOXP3 positive Treg cells. And section D shows that the phenotypic makeup of the Tregs, there are more than 90%, or actually more than 95% CD4 positive, CD25 positive, and FOXP3 positive cells, and there were very little contamination with non-Treg cells like CD8 positive T cells, CD20 positive B cells, or CD127 positive activated T cells. Next slide, please. This slide that shows how we functionally tested the Tregs in an MLR inhibition assay. Recipient responder PBMC were on the left-hand side. The recipient PBMC were stimulated with donor irradiated PBMC. To this MLR, we added different numbers of serial dilutions of expanded Tregs and control cells. After 7 days, thymidine incorporation assay was performed, and the data were calculated as percentage inhibition shown by the formula at the bottom left. As you can see in the graph, the Tregs inhibited the MLR very potently and in a dose-dependent manner. Since the assay results could be obtained only after 7 days, the 14-day MLR inhibition results were used as in-process testing. Next slide, please. For the trial, the release criteria included these aspects. Essentially, what it showed was we had good cellular viability. There was no bacterial, fungal, mycoplasma, or endotoxin contamination. For the polyclonal expansion, we used CD3 and CD28 coated beads. And so we had to remove those as per the protocol, the IND protocol approved by the FDA, and we can have only 300 or 3,000 beads per 100 million cells, and we removed those beads. And also, we found that these cells had excellent functional capabilities, as we had shown, as we had seen in the previous slide. Next slide, please. When we performed limited peripheral blood immunophenotyping by flow cytometric analysis, the most important finding is shown in this slide. We observed a change in the percentage of Tregs when serially followed up. When we consider the pre-transplant, percentage of Treg value as 1, what was the fold increase in the Treg after the infusion? We found, as you can see, that in the graphs, that there was a tremendous increase after the infusion. This was somewhat stable over time, and the patients who received higher numbers of Tregs tended to show increased amplification of Tregs in their blood after the infusion. Next slide, please. Another example. During the last 4 years, we have developed a technique for the generation and characterization of alloantigen-specific Tregs for clinical transplant tolerance. The basic protocol was similar to what I had mentioned just before in the polyclonal expansion. But the main difference was we had used donor-activated and irradiated B cells for initial stimulation. Now we are transferring this technology to the GMP. It is our expectation that we will need lower number of Tregs for tolerance induction as the cells are more specific and targeted. Next slide, please. And to summarize, Immune Monitoring Core can provide consultation and support services for developing cellular therapeutics for various steps during the product development, authentication, and quality assessment. Most importantly, we can provide serial immune monitoring during the clinical trial. I have a sneaking suspicion I did not describe one of the important slides very well. This one. Yeah, here I had mentioned only about the consultation, protocol development, and the assay development. Then I need to say that we will transfer the technology to the GMP, where we can have scaled up production of the product to determine feasibility and further modification of the protocol development before the clinical scale production, and then develop detailed SOPs for each of the steps in the product manufacturing. Then write an IND protocol after running several dry runs of the manufacturing product, and then submit an IND to the FDA and get permission to obtain IND to complete a clinical trial. So this I think I missed in my description. I'm sorry about that. Do you have any questions for me?
I haven't had any questions come through the Q&A box. So if you have questions, there should be a box in your WebEx portal where you can enter those. So to keep us on track with timing, we'll just move on to Dr. Vanoy, who's an expert in Northwestern's Department of Pharmacology on ion channel biophysics, structure-function relationships, and regulation. And he's here to tell you more about how his group can help your research with high-throughput functional annotation and pharmacological profiling of ion channels. So Dr. Vanoy, I will let you take it away.
Okay, can you hear me?
Yes.
Okay. Hi. Thank you for the kind introduction. Good afternoon. It is a pleasure to show you some of the work we do as examples of how it can be applied to your studies. Let me see if I have good control of the slides. Yes, I do. Our group in the Department of Pharmacology uses high-capacity, high-efficiency electroporation to achieve transfection of wild-type and disease-associated ion channels alone or with accessory subunits at sufficiently high levels for efficient automated patch clamp recording. This approach allows for high-throughput automated patch clamp to speed up the functional annotation and pharmacological profiling of ion channel variants. For efficient analysis of ion channel proteins, we have developed a workflow that combines high-efficiency electroporation using the MaxCyte system shown on the left, which allows for transfection of up to 40 million cells per electroporation, and high-throughput functional analysis by automated patch clamp. Our system is the SynchroPatch 768PE, shown on the right, where we can record from 384 to 768 cells per run. We usually do transient co-expression of ion channel proteins into heterologous expression systems, for example, HEK293T cells, CHO-K1 cells, or ND-23 cells. The cDNAs are engineered into vectors which also code for a fluorescent protein, which serves as a marker for transfection efficiency. Now, these are not fusion proteins, so the fluorescent protein is independent of the desired protein. We have also successfully generated cell lines extensively expressing one or two proteins, which now we can use as for experiments or also as expression systems for transfection of other proteins. The efficiency of electroporation, that is, the transfection efficiency, is then evaluated by flow cytometry using the Cytoflex benchtop flow cytometer. And that's in the next slide. Sorry about that. So on the left, we have the electroporation system. Once we do the electroporation, we can measure the efficiency of this electroporation by the benchtop flow cytometer shown on the right. This slide shows the transient co-expression of the voltage-gated potassium channel casein Q1 plus its accessory subunit KCNE1 following co-electroporation of the CHO-K cells with casein Q1 iris EGFP vector shown on the right plus KCNE1 iris DS red vector shown on the left. This allows us to measure fluorescence of green protein as a proxy for casein Q1 expression and of red protein as a proxy for KCNE1 expression. Therefore, we can follow the expression of both proteins independently into the cells that we have transfected. The plots below show the transfection efficiency for both plasmids evaluated by flow cytometry. We have currently getting levels of about 90% for one of the proteins for the channel protein, while we're getting close to 90% for the accessory protein. This is a typical experiment that we do. First, number one here on the left, the top far left, DNAs are after have been generated and purified using endotoxin-free MaxiPrep kit, are also fully sequenced to make sure that there are no mutations introduced during the generation, that is growth of DNA in the bacteria, or during the mutations that we have also introduced to prevent mutations that were not designed. Following that, we confirm that the DNA is of good quality, electroporation is performed, then the cells are incubated for 48 hours, and after that, we measure the flow by flow cytometry, the transfection efficiency. If the transfection efficiency is deemed high enough for subsequent studies, then we cryopreserve multiple vials. This allows us to do multiple experiments using the same cells. The day before an experiment, the cells are thawed, and they're grown at either at 37 degrees or 28 degrees for 24 hours. The day of the experiment, the flow cytometry is performed again to confirm that the expression levels of the desired proteins are still on target. And then finally, we do whole cell recordings in the automated patch clamp platform, where Here, we can also, while we're doing the experiments, we can expose the cells to multiple external or internal solutions to generate a complex matrix of experimental conditions. This slide and the next 6 are examples of what type of assays and results you can obtain by using our system, the system that we have. This is an example, this figure here, of an automated patch clamp recording of whole cell currents before and after adding a compound. This recording is from CHO cells transiently expressing KCNQ1 plus KCNE, which form the IKS channel, which is critical for cardiac action potential repolarization. In each panel, on the far left, are recordings from wild-type channels, followed by 5 distinct transfections of 4 mutants and also as a negative control non-transfected cells. On the left, we have the currents measured before adding the blocker, and on the right, we have the currents from the same cells measured after adding the blocker. These are examples from a recently published study designed to determine the functional properties of 48 KCNQ1 channel variants of unknown significance associated with arrhythmia, specifically long QT syndrome. This slide shows the average whole cell currents recorded from CHO-K1 cells co-electroporated with KCNE1 plus KCNQ1 variant. In this study, we also determined the functional properties of the KCNQ1 variants recorded by automated patch clamp, and here we show those results as changes relative to the wild-type protein. On the left, we have the activity level for each variant relative to wild-type channel, shown in a waterfall plot ranging from loss of function on the top to activity larger than wild-type on the bottom. On the right, we have the changes in voltage dependence of activation relative to wild-type for those variants with measurable currents, and those variants are shown by the solid lines depicting which mutants were able to be measured. Another example of the type of results we can obtain are illustrated in the next 2 slides, which show the results from an ongoing study to determine the functional and pharmacological properties of 70 KCNQ2 channel variants associated with epilepsy. This figure shows the average whole cell currents recorded from wild-type and 5 other mutations, and we can show here that we can detect either loss of function, for example, on the T274M mutant, to changes in kinetics of the channel, illustrated by the mutation R207W. Here we show that we can also determine the pharmacology of the ion channels. This slide shows the effects of reticabine on wild-type and variant KCNQ2 plus KCNQ3 channels. The top channels are the dose-response curves for reticabine on wild-type channels, either on the current density, current amplitude, which is panel A, or the voltage dependence of activation, which is panel B. We have also looking at the effects of the mutations on the pharmacology of the channel. On the left, panel C, we show the effect of current density on variant KCNQ2 channels, and one of the mutants is not responsive like the other ones. While on panel D, we're showing that the mutations all respond equally to changes in voltage dependence of activation when reticabine is added. An interesting point of these last 2 panels is that not all of the variants respond to the compound in a similar fashion. The mutation changes the pharmacological profile of the channel protein, which may preclude a therapeutic compound from being effective. Lastly, I would like to let you know that in summary, we hope we can help you achieve a high volume, high efficiency transfection of proteins into cell expression systems. This does not have to be just ion channels. Our system will allow us to transfect any kind of protein that you will be interested on. We will not be able to provide you help with the functional analysis if we're doing automated patch clamp, but we can help you generate the cell lines that you can then do for other studies. The cells that can be generated by the transfection can be then used for assays, including automated patch clamp recording. A high-throughput automated patch clamp can be used to assess functional defects associated with mutations in renal ion channel proteins. And also, finally, that automated patch clamp can also be used to determine the pharmacological profiling of ion channel variants or to test specific compounds that you may have against specific ion channel targets. If you have any questions, I'll be happy to answer them now, or if not, please contact me by email and let me know if you have any questions, and I'll be happy to provide any answers. Thank you.
All right, thank you, presenters. We'll go ahead and open it up and take some questions now. Just a reminder, be sure to type your questions into the questions box in your control panel. If you have a preference as far as you want to speak your question, you can also type that in there and I can unmute you. I guess one helpful question that might be good for people to know is when is it best for people to approach you? At what point should they be in the development of their research idea or their project when they come talk to you and seek out a consultation at any of your centers?
Well, I can start off. I mean, I think early, really, because we have kind of a comprehensive approach from actually developing the molecules to being able to test them using some of the different resources within the core. So again, we've kind of presented just a sliver of the opportunities available. And so if you're designing a small molecule or in the process of doing that, you can reach out to us and we can put you in contact with specific navigators, many of which you've seen today, that can help from the development of the concept of the chemistry of the small molecules, progressing on to determining PK/PD, or developing the cellular products, and then eventually transitioning into Core C, which would be more human applications. So the message is early, really, from concept, and we can help you all the way through.
Okay, great. Well, it looks like we don't have any other questions for now, but if you do have any other questions, you can reach out to us. We are on Twitter at NU Nephrology. You can also send us an email at nephrohub@northwestern.edu, and we'll make sure that your question gets directed to the core navigator that you're looking for. Or you can visit us at nephrohub.org and sign up for our emailing list to stay up to date on upcoming webinars, guest speakers, and any funding opportunities, and also learn more about our other resources and services. Again, just as a refresher, that nephrohub.org is also a place where you can get started with NU GoKidney Core, any of their cores, but in particular Core B, and get more details on the services that they offer. So keep an eye out for the recording and survey from this webinar, and thank you again for joining us today. We really appreciate your time and attention, and we'll see you next time.